Dear friends, today we are going to discuss some fundamental aspects regarding the morphology and distribution of arthropoda. Arthropods are the most diverse group of fossils and living invertebrate animals. They have colonized land, sea and air due to a relatively plastic and adaptable body-plan organization. Many different phyla of metazoans, including the arthropods, show a repetition of structures along their long axis. This is generally termed as metamerism. Arthropod bodies are characteristically segmented, and the different major groups are defined by various patterns, what you call as the tagmosis, which is meant by segment fusion to form heads, bodies and tails. The phylum derives its name from the characteristic jointed appendages found in all the members, and it is derived from the Greek word aros or arthros means jointed and podos means foot. These jointed appendages, which consists of individual elements called as the podomeres, which take the form of walking legs, gill, branches or antenna. Broad divisions can be made within the arthropods depending on the exact nature of the leg appendages. Uniramians or arthropods with single branched appendages retain only a walking and swimming leg. Biramians have an appendage which consists of two separate elements, a leg branch for a movement and gill branch used in respiration. That is exoskeleton. All the arthropods possesses a rigid exoskeleton which provides support and protection and acts as a buffer between the external environment and internal body process. The exoskeleton of arthropods is composed of a carbohydrate polymer, what you call as the chitin. In some arthropod groups, the chitin superstructure is reinforced with mineral salts such as calcium carbonate or calcium phosphate. Arthropods with this biomineralization show a higher preservation potential than non-mineralized forms and consequently have a more complete fossil record through time. For non-mineralized forms, we have to look to sites of exceptional preservation, that is the fossil conservation or lagersta, to trace their evolutionary history and discover aspects of their paleobiology. Localities such as Middle Cambrian Burgess Shale, the Ordovician Somme Shale, the Devonian Rhynie Chert, the Carboniferous Mason Creek Nodule Hosted Biota, the Jurassic Solnhofen lithographic limestone, and Baltic, Dominican and Mexican ambers all provide windows on to the arthropod faunas at stratigraphical points in geological time in which preservational failure would otherwise obscure our view of evolution of the various groups. Furthermore, there is a gradation of fidelity of preservation even in these special examples. To this end, it is entirely possible to study the exquisite three-dimensional outer surfaces of tertiary flies in the amber or the internalized respiratory systems of Devonian arachnids. A consequence of the possession of a rigid exoskeleton composed of cuticle is that in order for the arthropod to increase in body size over time, the rigid cuticle has to be shed or molted and a new cuticle grown below the surface to replace it. Arthropods differ from many other animal groups in that they go through the process of ecdysis, that is literally shedding the exoskeleton as a mold in order to attain greater body size. Ecdysis is facilitated by the presence of sutures in the exoskeleton, lines of weaknesses which can be broken through so that the arthropoda struggle free of the old skin. The ontogeny of ecdysis allows segment adhesion and tagmosis to be identified from the first instars right up to the adult forms. Unfortunately, for arthropods, molting of the old skin and the time taken to harden the new exoskeleton leaves them vulnerable to predator attack or adverse environmental conditions. For this reason, many arthropods, both in aquatic and terrestrial environments, seek out a molting refugia in which the process of ecdysis and subsequent hardening of the new exoskeleton can take place in relative safety. Where mortality occurs during this post-molt stage, the cuticle of the arthropod fossil formed can display wrinkles or even tears in the structure. The cuticle itself takes the form of the hardened sclerites, 1% or 1 per body segment. Dorsal sclerites are termed as tergites, ventral sclerites are termed as sternites. Where the tergites and sternites are fused together, they form a simple ring-like structure enclosing the arthropod soft tissue. Lying between these discrete segments are the areas of connective and unmineralized cuticle. The presence of the less robust construction material explains why upon death well-mineralized arthropods such as trilobites can disarticulate to a greater or lesser degree into their constituent parts. The leg appendages in arthropods are similarly constructed with cylindrical reinforced cuticle produmeres with intervening less well sclerotized arthrodial membranes connecting them. Muscle attachments required to move the individual leg podomeres are attached to small boss-like projections from the inside of the exoskeleton called as apodemes. The cuticle of the arthropods explains in part their success in the colonization of such a wide variety of different environments with widely varying challenges. The cuticle provides a physical barrier between the internal organs and the external environment, which helps to reduce desiccation through water loss in terrestrial forms, and a chemical barrier in aquatic conditions where hypo or hypersalinity could upset the osmotic balance, leading to dehydration of the organism and ultimately leading to the death also, as well as a physical barrier waxes produced by the cuticle layer help to waterproof this material. Some other more unusual properties of arthropod cuticle have been identified recently, including the fluorescence under ultraviolet light of the hairline cuticle within the exoskeleton of scorpions. It happens that this hairline layer is actually very resistant to the bacterial degradation and is therefore often the main remaining constituent material of fossil scorpions. For example, the early Carboniferous fast Crichton biota of West Lothian, Scotland contained a number of different scorpion taxa which were recovered by hydrofluoric acid maceration of the enclosing limestone matrix. The level of morphological detail made available through this technique was as good as the studying extant species. Classification schemes. There still remains a scope for a significant disagreement in the higher level taxonomy of this group of animals. Historically, the main dichotomy relates to whether the phylum Arthropoda is a monophyletic or a polyphyletic group of organisms. Some variation occurs in the higher level classification of this group, with some authors recognizing Arthropoda at phylum level and the constituent members variously as subphyla orders. A monophyletic grouping for the arthropods would imply that all of the lower level groupings originated from a single common ancestor. This was the largely historical viewpoint based on the striking common appearance of the various groups. In order to counter what seems to be logical conclusion based on many shared characters in common, the argument for polyphily has to promote plausible explanations for the observed similarities between groups as well as providing proof of separate points of origin. The fossil record may provide just such evidence, allowing the evolution of the various arthropods to be considered with the benefit of deep time hindsight. However, since the work of the Sidney Manton, who suggested that three major groups, the Crustacea, the Chelicerata, and the Uniramia, would more comfortably be accommodated as phyla of their own, the consensus view has swung back to the likelihood of the monophyletic status of the group being the correct conclusion. This has been due in part to the advent of molecular sequencing techniques. A novel or non-morphological hypothesis linking crustaceans and hexapods were primarily based on the analysis of single or a handful of the genes. However, as the science advances and the techniques of the extraction improves, the case of monophily appears to be more solid. Indeed, one study not only supports the crustacean hexapod clade but also recognizes pycnogonids as the sister group to other euarthropods. The historical convention has been to subdivide the arthropoda into the following equal biased groupings, that is Trilobita, containing all the trilobites but excluding narroid arthropods, Chelicerata, that is Arachnida, Scorpionida, Xiphoceura, Eurypterida, Chasmatapsida, Crustacea, Uniramia, including both insects and myriapods. However, as more and more examples of basal arthropods and oddball taxa are discovered, primarily from the fossil conservation Lagersta than other, less conventional higher taxon groupings can be identified. Some examples include the Atelocerata, that is the Diplopoda, Chilopoda, Apauropoda, Symphyla hexapoda, Arachnomorpha, that is Arachniata, trilobites, chelicerates, Trilobitomorpha. Arachnidomorpha is a clade which basically consists of all the taxa more closely related to the chelicerates than crustaceans. Panarthropoda is again a clade, but this time containing all true arthropods and their soft-bodied relatives, such as the Onchophorans and the Tardigrades. To this end, the term arthropod should probably be used to represent a grade of organization in which a soft-bodied ancestor, presumably a segmented form, developed a tough or toughened cuticle. Gene sequencing and molecular markers may provide fascinating new insights into the interrelationship of the various high-level arthropod groups. What the fossil record says. Tracing arthropods back through geological time indicates that the division between the major groups extend almost as far back as the fossil record of metazoa, convergence of form and channeling bifunctional morphology of multiple arthropod or arthropodization segments through forming a rigid cuticle could have led to polyphyly. However, the more complete our record of the group becomes, the less likely this seems. The first arthropods appear in the fossil record of life on Earth in the Lower Cambrian some 540 million years. The arthropod faunas of the Xinjiang biota of China demonstrate that even at this early stage, a diverse range of arthropod body plans had evolved. By Middle Cambrian times, rapid and explosive radiation and evolution of arthropods led to the development of many different body plans, including the majority of those still extant. This Cambrian explosion in the number of and diversity of arthropod around plants was one of the first major radiations of the group and took place in the marine environment only. By the Upper Cambrian, trilobites had diversified markedly, and this carried through into the Ordovician times. Mid-Silurian and Lower Devonian rocks yield evidence of the next major step in the evolution of the arthropods. The colonization of the terrestrial environment, a process termed as terrestrialization. Different terrestrial arthropod groups probably took different terrestrialization pathways, although this is difficult to determine from the fossil forms. Extant members of the ancient lineages can lend clues as to what these pathways might have been. For example, osmotic concentrations of the body fluids in Maripods suggest that this group terrestrialized and straight from the sea without an intermediate step directly into the fresh water. One particular locality which has yielded disproportionately large amounts of data on early terrestrial ecostyles of the ecosystems is the Devonian Rhynie chert Lagersta in the Aberdeenshire, Scotland. Here, the silica mineralizing action of hot spring and the geyser fluids captured in three-dimensional detail early terrestrial arthropods such as arachnids, insects and centipedes, as well as aquatic forms also, such as freshwater crustaceans and euthyrcanoids also. The remarkable morphological stasis in early terrestrial arthropods in relation to the extant forms suggests that the arthropod-dominated terrestrial ecosystems stabilized early on the Earth's history, and that soil and later habitats and inhabitants have changed very little ever since. One of the most novel innovations of arthropods was the evolution of muscle-powered flight in the insects. The earliest insects, or apterygotes, that is insects without wings, first appear in sediments of Devonian age in the form of Colombo sandicenturans. In earliest Carboniferous times, the major expansion and evolution of land-based flora provided new ecological niches available to this highly adaptable group. The advent of insects and the development of the flight led to the colonization of an entirely new ecospace. Interestingly, the earliest non-scorpion arachnids appear to have relied solely on the poison fangs to capture and subdue prey items. Orb weaving using a spider silk could only have developed in response to the presence of aerial insects. By lower Carboniferous times, the first winged insects had developed, and an explosion in the diversity of insects occurred due to coal swamp forest ecosystems. Preservation and discovery are aided by the presence of siderite concretions in the roof shales to many coal seams, which in turn are commercially exploitable. During the Middle Carboniferous, insects became diverse and widespread for the first time in their fossil record. This trend continued throughout the Carboniferous and into the Permian period also, whereupon an explosion in the total diversity of fossil insects took place. The depositional environments in which these fossils are found do not differ significantly. That suggests a real increase in the diversity. During the Jurassic and Cretaceous, favorable shallow epiric sea conditions saw the emergence and dominance of decapod crustaceans as a major player in the near-shore marine ecosystems. This development within the Crustacea continues to the present day, with arthropods still developing new and successive waves of colonization, particularly in terms of the terrestrialization of some decapod crustaceans, such as the land crab or other examples. Relevance. Arthropod fossils are used by the paleontologists and geologists in a number of different ways for different reasons. In rocks of Paleozoic age, the trilobites have long been used as the biostratigraphical marker fossils. They find application in this particular field of study as they are readily fossilized. Carbonate impregnated exoskeleton, numerically abundant, each individual molds many times over a lifetime, amplifying their number of potential fossils, and show sufficiently rapid speciation events to make them useful index fossils. In early Paleozoic rocks, nectonic species are useful in paleogeographic reconstructions of the former oceans, seas, and by default land masses also. The nectonic lifestyle has led to a widespread occurrence of the resultant fossil remains, ideal for the broad stratigraphical correlations. Another use for the numerically abundant trilobites in stratigraphical framework is the evaluation of evolutionary rates within well-defined lineages. Such frameworks allow trilobites to be utilized in studies of evolutionary changes within a species, up until the point at which a new species evolves, and a field of study known as the microevolution. Finally, many micropaleontological samples such as the fossils of ostracods are similarly numerically abundant and readily preserved and have found application in paleontological archaeological studies of past environments due to their environmental sensitivity. The calcium carbonate impregnated shells of these small bivalve carapaced crustaceans are a common constituent in the rocks of all ages from the Cambrian onwards. Coming to the visual systems. Two main types of eyes are recognized in the arthropods, a compound, that is multifaceted, and simple ocellular eyes. Compound eyes probably developed separately in a number of different arthropod groups, giving rise to analogues if not homologous structures. Many arthropods possesses both compound and simple eyes, as demonstrated by the extant chelicerate horses or horseshoe crab lumulus. The ocellular eyes are positioned on the top of the dorsal surface of carapace and are primarily used to detect light levels, indicating whether or not the top of the carapace is covered by sand. This information is of particular use to the burrowing arthropods. The compound eyes of the trilobites are further subdivided into holocoral and schizocoral forms, with the schizocoral structure developing as a result of pedomorphosis. Holocoral trilobite eyes consist of many round or polygonal lenses, the edges of which are all in contact and covered by a single corneal membrane. Schizocoral eyes are a unique system restricted to certain trilobite groups, that is the Phacopina. The lenses are large and separated from one another. Each separate lens has its own separate corneal covering, mounting the eye lens on under or upstanding, and curved turrets may have allowed certain trilobite species a full 360-degree field of vision. Coming to the trace fossils. The combination of rigid appendages and the ability to be motile in a wide range of ecological habitats has provided a rich and diverse trace fossil record which can be assigned to the activities of arthropods. Trace fossils can indicate the presence of arthropods in depositional environments in which preservation would normally fail and body fossils are non-existent. A good example of this is the Permian Cocosino sandstones, a formation of the Arizona. Various studies of the desert sandstones have revealed the presence of scorpion and tarantula trackways, the body fossils of which are absent from these environments. Distinctive trace fossils such as the Capuchinum can reveal the presence of Xiphosaurian arthropods, horseshoe crabs, and by analogy with their extant relatives such as Limulus can be used to employ nearshore or freshwater paleo environments in otherwise sedimentary or sediment devoid of body fossils. Trace fossils, when found in conjunction with the body fossil assemblages may allow behavior to be interpreted, such as the speed of movement or different actions, nesting for aging, etc. Of particular importance in the fossil record of insects is the formation of leaf mines and bite marks in accompanying floral assemblages, also giving a clear indication of an escalating arms race between plants and arthropods. Thank you very much.